Welcome and welcome back to my YouTube channel. So today we are going to discuss new chapter of first year class, which is enzymes. So yeah, first of all we're gonna look at the enzyme. The enzyme was given by the scientist named as Friedrich William Kuh. He basically proposed these enzymes in 1878. The scientist and this date can be asked in most of the past papers, so I must learn this. After that, what actually are these enzymes? So enzymes are basically biocatalysts. Enzymes are what? Enzymes are biocatalysts. Then another point we can mention that enzymes are made up of what? So enzymes are made up of basically polypeptide chains. Perfect. Then after that, where these enzymes are formed. So these enzymes are basically formed in protoplasm of a cell. In protoplasm of cell. After that, is we're gonna discuss another point, which can be we can say so enzymes have high molecular weight. Sorry. Mm, yeah. So enzymes have high molecular weight. And this weight have a range, so it can be range from 10,000 to 1 million. Okay, then after this, these enzymes, as we have discussed in the protein chapter, that enzymes like proteins have two types. One is globular protein and second is fibrous protein. So these enzymes are what? These enzymes are globular protein. Globular protein. Now their structures look like either they are tertiary in structure or they are quaternary in structure. Quaternary. After that, we can say we do have RNA. As we all are familiar with RNA, what is RNA? It is ribonucleic acid. So there are some proteins of RNA or some RNA which also act as enzymes. Some proteins of RNA act as enzyme. Act as enzyme. So the proteins of RNA which act as enzyme, they are known as ribozymes. They are known as ribozymes. Ribozymes, right? After that, here one thing I forgot to mention the word basically enzyme meaning. So as we know, word N means inside. With that, zyme means yeast. Okay, N means enzyme means yeast. Then we do have basically the active side. We know that, like for suppose this is the enzyme structure. So this is somehow I have enzyme, and we know that with the enzyme the substrate molecule attached. So here I have three type of substrate. One is this, second is this, and third is somehow like this. So now we know that at a particular site, the active site is what? This is the active site of enzyme where actually the substrate attached. Active site where substrate attached. Okay. So now what happens? This enzyme will attach to its specific substrate. ये जो भी हमारे पास enzymes होते हैं, enzyme at the active site of enzyme, the particular we can say substrate will attach, which will be complementary to the shape of the active site. So we know that from these three, which is basically the complementary to the shape of active site. So of course this is the complementary to shape of the active site. So we're gonna attach this I'm going to remove this from here and I'm going to attach this over here. So yeah, this is how it attached to the active site of enzyme. Fine. Now here we know that if when this attached to the active site, so active site of enzyme is divided into two parts. This is active site of enzyme is divided into two parts. One part which can be mentioned over here. Let me change the color. Yep. Okay. So one part if I can mention here, that is binding site. Binding site. Another is what? Another is catalytic site. Catalytic site. Now active site, there are two sites. Now what is different between binding site and catalytic site? So yeah, here you can see some of the specific amino acids are present on the substrate which will directly attach to the active site. A specific amino acid bind to the active site. So this is known as binding site. But what happened in the catalytic site? So in the catalytic site here, known factor play a major role. Basically, it helps to convert substrate into product. Substrate into product. We know that the substrate converted into product and the amino acid binds. These both we can say role take place at the active site. ये दोनों हमारे पास active site पे होते हैं. But here you can see what binding site पे only amino acid bind. They do not start or they do not directly convert the substrate into product. But at the catalytic site of this active site, they convert substrate into product. They convert substrate into product. I hope till now it is clear. Now let us discuss about some of the major topic. Here we do have what? Here we do have protein plus non-protein part. We know that in enzyme we do have both part are present. So protein plus non-protein part, they basically both combine and form what? They both combine and form conjugate enzyme. Conjugated enzymes, right? Conjugated enzymes. Now the separate non-protein part is known as what? The separate let me write over here. Yeah, separate non-protein part is known as cofactor. Non-protein part is known as what? It is known as cofactor. Cofactor. Yep. Now here you can see this cofactor is again divided into two parts. Number one and second. So first part cofactor is basically it can be inorganic cofactor. Inorganic cofactor. Another can be organic cofactor. Organic cofactor. Now, what's this difference between inorganic cofactor and organic? So, basically, inorganic cofactor include metallic ions. Metallic ions, like we know, we do have magnesium, another chlorine. So these are what these basically in inorganic ions we can say these act as activators. These act as activators. They help to activate the enzyme function. So they help in the activator. They are activators, they help in the activation process. So now why these are inorganic? Because these are deattachable. These will not be attached at the time where substrate convert into product. These are deattachable. They can be go activate the reaction and then remove. So they are deattachable, right? Now here in the organic side, yes, here in the organic side we do have again two groups. Sorry. Yep, here we do have again two groups. Fine, number one group is coenzyme and second is prosthetic group. Right, these are two groups which are present, coenzyme, and second is prosthetic group. I can mark here. Yep. So now what's the difference between coenzyme and prosthetic group? So here you can see these coenzymes are what? These coenzymes are basically derivatives of vitamin like nicotinamide, FAD, NAD. These are derivatives of vitamins. Vitamins like NAD, FAD, and many others. These are attached to the protein. They are not detachable. However, these are attached. Okay. These are also known as what? These are, we can say these coenzymes are also known as inorganic. But we did not give the name to inorganic. We put that into the organic category because these are attachable, and we know inorganic are detachable, right? Now here in the prosthetic group we do have these. We can say cofactors are covalently and permanently bind to molecules. Covalently and permanently bind. They do not detach. However, they not attach. They are permanently binded. For example, we do have magnesium, which is present in the chlorophyll porphyrin ring. Chlorophyll porphyrin ring, which is attached, right? So this is known as prosthetic, and we do have these are the inorganic cofactors. Now here's the concept of haloenzymes and apoenzymes. Yeah, here the concept of haloenzymes. What are these haloenzymes? So let me mention here, haloenzymes are basically the polypeptide chain plus cofactor. When they both combine, they form holoenzyme. Polypeptide chain and cofactor is known as holoenzymes. And another is apoenzyme. What are these apoenzymes? So apoenzyme is nothing as difficult, but here we do only the protein part. Only protein is present, or we can say apoenzyme is the conjugate enzyme without cofactor. Conjugate, we know conjugate include protein plus non-protein part. So conjugate enzyme without non-protein. So without non-protein, this means it contains only protein part. Okay, without non-protein. And this was a basic concept of enzymes. If it is clear and if you have any question, you can drop the in the comment box. Now we are going to discuss about the mechanism of the mechanism of enzyme basically. Yep, here we are going to discuss mechanism. So we do have basically two models, which one was performed by induced model, another was performed by was lock and key model, right? Okay, so in the mechanism, we do have basically we know that what we know that substrate when combined to the active site of enzyme, they combine to active site of enzyme. But here the series of there's a proper arrangement how these actually combine. So let's discuss that arrangement. Okay, so let's discuss that arrangement. So first of all, we know that when substrate combine to active site of enzyme, they form what? Substrate enzyme complex. Or we can say don't write complex, however, but substrate enzyme. But here we can write substrate enzyme complex is formed. Then when the substrate enzyme complex is formed, now the reaction starts, and then substrate directly convert into product. But when the substrate is converting into the product, still the enzyme is present into it. Okay, so this means this will form um, enzyme EP, enzyme product complex. Enzyme product complex, because product is not completely formed. So that's why enzyme is still present. After this enzyme complex, when product is completely formed, they will remove the product. Product is removed and plus enzyme is also removed. We know that, so, and this has proved that enzyme do not take part in chemical reaction. Do not take part, not take part, we can say, do not remain before and after the chemical reaction. Remain after the reaction, it is removed. Okay, so this is basically the complete series which you have to remember. This is mostly being asked, so yeah, perfectly fine. Now we do have two models was given by, so one is lock and key model. Now here the main points to be noticed that lock and key model was given by Emil Fischer. With that, this basically model comprise that we know that particular key can be adjusted into the particular lock. For example, this is one type of lock, and in this lock the particular key can be fixed. So same as in the particular enzyme, the particular substrate can be fixed. This is basically the basic concept of lock and key model. This model basically tells us that active site of enzyme is what? Active site of enzyme is rigid, hard. So that's why the proper substrate can be attached to the active site according to that Wegener shape. Yep. Then another complex Wegener model can be which was induced state model. Now induced state model basically comprises that this was given by Koshland in 1959. Right, 1958 or 9 maybe. I don't exactly remember, but 1958 or 1959, you may check. Then, and this model basically says that, yeah, here we can say active site is flexible. Active site is flexible. So there are some enzymes, some substrates which are not according to that enzyme, according to that shape, which can be easily attached to active site. But the substrate change the active site, alter the shape of active site, change the shape of active site. So when the shape of active site is changed, it is according to any substrate molecule. So we know that the product will be formed, but not exact product that we need. Okay, because here active site is flexible, it can be easily adjusted according to any substrate shape. So this was all for today. In the next video, we're gonna discuss about some of the factors and then another some of the basic terms with that plus MCQs. Till that, bye bye.